Chapter 8, Section 3, Cell Division and FTS Proteins. Now you've made the copy of the chromosome. You need to divide the cell. How do you do that? Well, you have what's referred to as the divisome. Okay, this is the proteins that are literally going to divide the cell in half. Well, maybe not in half, just divide it, depending upon the cell. Could be in half, could be just a small part, like we saw last section with Caulobacter. A lot of this is going to be under the control of what are known as the FTS proteins. FITZ is going to be our crucial one. This is the one, remember back in the first section of this chapter, I showed you the ring, that is going to be that contractile ring. That was primarily composed of FITZ. FITZ is a tubulin-like protein. Remember, tubulin is our microtubule cytoskeleton in eukaryotic cells. It's the big tube-like cytoskeletal component. So this FITZ tubulin-like protein is going to form that contractile ring that's going to pull in the cytoplasmic membrane and the peptidoglycan layer. There are going to be other FTS proteins that we're talking about. Remember, these are going to be bacterial only. Archaea is going to have their own version, and the eukaryotic cells have a completely different set of proteins, depending upon whether they're fungi, plant, or animalia. It's easier to talk about this originally in rod-shaped, like E. coli, the Bacillus E. coli. Okay, because you're going to get FITZ at the center of the cell. It's going to form a ring that interacts with FITZ and ZipA. ZipA, this is what anchors FITZ to the cytoplasmic membrane. So this is going to be an integral protein inserted in the cytoplasmic membrane that is interacting with FITZ. FITZ is an actin protein. It's an actin-like protein. This recruits FITZ and the other proteins to the center of the cell. So when you look at them here, okay, ZipA is the integral protein that anchors FITZ, here's part of the contractile ring, anchors it to the cytoplasmic membrane. FITZ recruits in everybody. FITZ, FTS, FITZ-I, FITZ-K, I, K, and a few others that aren't shown here. Their jobs are to help remodel the cytoplasmic membrane and the remodeling of the peptidoglycan layer. You look here in B, you can see the setting up of this FITZ ring. And as the ring gets there and it contracts down, you get alteration of the envelope where it pinches in. And then you'll ultimately end up with two independent cells. Now, this contractile ring, this divisome. Wow, I cannot say words today. This divisome, the FITZ ring and all of that, doesn't start off there at the beginning of this process. It comes late to the party because there's so much there. It would interfere with movement and the separation of the chromosomes, so it comes later. When you look at the FITZ proteins there, remember the FITZ-I? Well, they call this the penicillin binding protein. Not that its job is to bind penicillin, it's just that they found that all of this can be inhibited, this whole process can be inhibited in the presence of penicillin, and they found that the penicillin binds FITZ-I, and that's why they called it that. All of these FITZ proteins are there for the peptidoglycan synthesis and the remodeling. Interfere with FITZ-I, well, none of this occurs. The remodeling of the peptidoglycan cannot happen. As the cytoplasmic membrane being pulled in by FITZ-Z can only pull in so far, it can only draw in so far before the membranes, any membranes will tear. So they have to replenish, add in new phospholipids. They're going to be adding it in so that as that FITZ-Z ring contracts in, pulls in, new phospholipids, more membrane. You get the remodeling of the peptidoglycan layer, and before you know it, you have new cell wall division by a septum. As I said, the DNA replicates before FITZ-Z ring even forms, okay? So there is this, timing factor here. There are proteins, there are metabolic processes that as the chromosomes, the nucleoids are copying, and then you see them segregating, separating to different ends. The FITZ-Z ring, it's physically inhibited from forming. Once this has completed, you're going to see a collection of proteins. Min-C, Min-D, Min-E will complex together and then will position FITZ-Z at the midpoint. FITZ-K and some other proteins will ensure that the chromosomes are separated and have been segregated to what will become the new septum when that cell wall forms. FITZ-Z depolarizes, starts to trigger an inward growth of materials. So it's not so much, as I said, a contractile pulling in, it's depolarizing, outer pressures pushing in as things reform around it, and then the FITZ-Z ring just kind of shortens and shortens. So it's a bit of both, pulling in by FITZ-Z, but then the inward growth of the peptidoglycan is also accelerating the movement in. This is a GTP, guided event. So when you look at what's happening here, remember the Min-C, Min-D, Min-E, they aid in separation of the chromosomes. Once the chromosomes have been pulled apart to a certain point, not complete yet, the Min-CDE complex will position the FITZ-Z ring. FITZ-Z, FITZ-A, ZIP-A, all of that. You get the depolarization, which causes remodeling of the peptidoglycan layer. So instead of being a continuous this way, that remodeling now makes it start to dip in right in the area of FITZ-Z. So it starts pushing in, and you got the FITZ-Z foreshortening. Because remember, it's a bunch of tubulin. Just keep knocking out the tubulins, and the ring gets smaller and smaller. And before you know it, the remodeled peptidoglycan comes into contact with each other. And the membranes either tear or reform as two complete cytoplasmic membranes. And then before you know it, the peptidoglycan layers will separate and the outer membranes will reform.